For almost all of human history, the code that makes us who we are, our DNA, was just a given. It was seen as permanent, totally unchangeable. But what if it wasn't? What if we could actually go in and edit the instruction manual of life itself? Well, today we're diving deep into the CRISPR revolution. And I want to be clear, this is not science fiction anymore. We are talking about a real technology that gives us the power to rewrite DNA, the fundamental code that builds every single living thing on this planet. The stakes, they honestly could not be higher. Okay, so let's get into it. And maybe the most incredible part of this whole story is that CRISPR wasn't just invented in a lab from scratch. No, it was discovered. It was hiding in plain sight, inside this ancient, microscopic war between bacteria and viruses. Now, the journey from just a scientific curiosity to a tool that can change the world, it took decades. It all started back in 1987. With this little mystery, scientists in Japan noticed these really weird, repeating patterns in bacterial DNA. In fact, that's where the name comes from. CRISPR is just an acronym for Clustered Regularly Interspaced Short Palindromic Repeats, which is a super technical way of describing those patterns. It wasn't until 2012 that Jennifer Doudna and Emmanuel Charpentier finally figured out what it was for, a programmable defense system for bacteria. And they realized, hey, we can harness this. By 2020, their breakthrough was officially recognized with a Nobel Prize. So, how in the world does this thing actually work? It sounds incredibly complicated, right? But the basic idea is, well, it's surprisingly elegant. So let's break down how you actually go about editing the most fundamental code in biology. Let's forget all the complex molecular biology for just a second. The absolute best way to think about CRISPR is like a pair of tiny, tiny molecular scissors, or maybe a scalpel, with its own built-in GPS. That's really the core of it. It can navigate the entire vast map of a genome and make a precise cut at the exact spot you tell it to. It all boils down to 3 key steps. First up, search. A little molecule called a guide RNA acts kind of like a heat-seeking missile, homing in on the one specific genetic sequence we want to change. Once it locks on, we're on to step 2, cut. A protein called Cas9, that's our molecular scissors, comes in and snips the DNA right there. And then step 3, repair. Your cell's natural instincts kick in, and it rushes to fix the break. And this is where the magic happens. Scientists can hijack that natural repair process to either shut off a bad gene or, even better, paste in a new correct piece of code. And that is why CRISPR changed absolutely everything. It's not like gene editing didn't exist before. It did, but it was just painfully difficult. We're talking slow, ridiculously expensive, and honestly, pretty unreliable. Then CRISPR comes along and just blows the doors off, making it fast, cheap, and incredibly precise. The whole field just opened up overnight. And this is where the story makes a huge leap, from the lab bench into the real world. We're not just talking about a cool research tool anymore. CRISPR is now out there, actively changing and, yes, saving human lives. This is the year it all became real for patients. You're going to want to remember this date because it marks a massive turning point in the history of medicine. In 2023, the FDA gave the official green light to Casgevy. It's the very first approved medical treatment built on CRISPR technology. All those decades of research, all the hope, all the debate, it all led to this one moment. Gene editing was no longer a what-if. It was officially a therapy. And just look at what this means for people. For patients who have been suffering from these awful inherited blood disorders, like sickle cell disease, doctors can now literally take out their stem cells, use CRISPR to fix the faulty gene, and then put those corrected cells back into their bodies. And the result? For some people, the constant pain, the debilitating symptoms that they've lived with their entire lives, just disappeared. This isn't just treating a disease, it's fixing it at its absolute source. But the story of CRISPR isn't just one of medical miracles. Oh no, it is also a massive cautionary tale. Because just as the world was celebrating this incredible therapeutic promise, a scientist on the other side of the planet took a step that just shocked everyone. 5 years before that first cure was approved, a line was crossed. And in 2018, the entire conversation around CRISPR changed forever. So a scientist named He-Jin Koo drops this bombshell of an announcement. He had edited the genes of human embryos, and what's more, twin girls had been born from them. He had targeted a gene called CCR5, and his goal was to give them resistance to HIV. Well, the reaction was immediate, and it was universal. Pure shock, outrage, and condemnation from the entire global scientific community. It ended with him in prison. A sacred line had been breached. And this, right here, is the critical difference you have to understand. That approved therapy, Casgevy, it uses what's called somatic editing. It fixes cells in one person's body. Those changes live and die with that person. It is medicine.
What He Jiankui did was germline editing. He edited embryos. That means the changes he made are permanent and can be passed down to every future generation. He didn't just alter two lives, he altered the human gene pool itself. So that controversy just threw us headfirst into some of the deepest, most profound questions our species has ever had to face. I mean, now that we have this incredible power, what do we actually do with it? How do you even begin to control the ability to shape our own evolutionary path? And this is the heart of the dilemma right here. Pretty much everyone agrees that using CRISPR to cure terrible diseases is a good thing. But what about using it for enhancement, to design certain traits in our children? That is where the consensus just completely shatters. Okay, now, before we all get carried away with sci-fi movie scenarios, let's pump the brakes and look at the actual science. The whole designer baby idea, it is way more complicated than it sounds. Traits like, say, intelligence aren't controlled by one gene you can just flip on. They're polygenic. They're influenced by hundreds, maybe thousands of genes all interacting with each other and with the environment. There's no single intelligence gene to go in and edit. And here's the thing. Even as we're all debating the ethics, the technology itself is not standing still, not even close. Scientists are already developing the next generation of these tools. Things like base editing and prime editing, they're even more precise. They work less like scissors and more like the find and replace function in a word processor, making changes with way fewer errors. The tool is just getting more and more powerful by the day. Just let that one sink in for a minute. For billions of years, evolution was this blind process, right? Random mutation, natural selection. Well, that era is over. We are now the very first species on this planet that can consciously, deliberately direct its own genetic future. So in the end, the question isn't, can we do this anymore? The technology is here. The real question, the one we all have to face now, is much, much bigger. Now that we're the ones holding the pen, what kind of future do we want to write?